Understanding mucus is key to understanding digestion. In the stomach, the lining has numerous gastric pits that include chief cells that produce pepsinogen, the precursor to pepsin, and I'll just do as a reminder C-P, and parietal cells that produce hydrochloric acid, and I'll refer to them as PHCl. So these are secreting hydrochloric acid, which of course dissociates into a proton, and chloride is a strong acid. Chief cells are secreting pepsinogen, which I'll call PG. A large number of the cells in the gastric pits and on the surface secrete mucus, and they secrete mucus in two forms. One form is tethered to the surface of each cell. That form produces an immediate barrier. So the pepsinogen and the acid that are leaving the gastric glands and the gastric pits are not able to get at the individual cells because of this layer of mucus that's tethered to the surface of the cell. They also produce a non-tethered mucus that forms either long strands or crosslinks. And so you wind up with a layer of mucus that includes tethered mucuses at the bottom and then free mucus in a gel. The mucuses resist penetration by protons. Protons can penetrate small distances down through them. The pH here may be very acid, pH 3 or lower. The pH down by the surface of each cell is close to 7.0. The stomach is able to produce this highly acidic fluid with high numbers of protons and pepsinogen. Acid will break off part of the pepsinogen protein, leaving behind pepsin. The small number of slowly liberated pepsins, liberated by acid, each can then act on pepsinogen. Active pepsin can now start breaking down proteins. Parietal cells produce hydrochloric acid that moves out past this mucus layer. Cells are protected from the hydrochloric acid by tethered and free mucus. Chief cells release pepsinogen, which is an inactive zymogen, activated the first few by acid, and then most of the rest by the free pepsin to produce pepsin, providing an environment in the stomach that breaks down food, is very hostile to microorganisms, and yet still has this protective layer of mucus so that the cells are not rapidly killed. Nonetheless, the stomach is still a hostile environment, and these cells are constantly being replaced from stem cells at the base and have an average lifespan in mammals that typically is measured in days. I want to examine mucus production in a little bit more detail. In the stomach, there are two mucus layers. The outer mucus layer is constantly being shed and is more likely to have bacteria. The inner mucus layer includes, at the base, mucuses that are tethered to the cells and more structured mucus up above it. There are multiple mucus-secreting cells here, Mucin 5AC. If we looked closely at the mucus in individual cells, here's a single cell with microvilli, and out at the tip, we're looking at a small section. So here's the cell membrane and mucins here. The mucin contains a membrane portion that's embedded in the membrane and a portion that extends out into the extracellular space in the stomach or the intestine with different patterns of glycosylation, and that's part of what gives different mucuses their different functions. The point of these mucus layers is to protect the cells of the stomach and the intestine from, for example, in the stomach, the acid and pepsin that would otherwise destroy these cells. The mucus also helps to hold a sodium bicarbonate layer, and this bicarbonate helps to protect the cells from acids that will combine with HCO3- to form carbonic acid. In the intestine, the mucus serves to keep bacteria and other living organisms in the gut away from the cell. Few bacteria penetrate. Some pathogens do burrow through the mucus and reach the cells. Here are two genes. The MUC1 here is showing this membrane-bound component with a cytoplasmic and an extracellular component, N-glycosylation, and O-glycosylation, these long strands. These interact extremely well with water, so once released from vesicles, they expand with water to 100 or 1,000 times their volume. When mucins are expressed, there are two forms. There's the cell surface mucins here I've just shown, and there are also secreted mucins in red. They're glycosylated. They can be dimerized. Here the two are dimerized, and again they're also glycosylated. They're packed into granules, so they're water-free. So once they're released, they expand 100-fold. A very small amount of material forms a large volume. Some are attached to the membrane and others are secreted. The secreted ones can form long tangled strands. Here are all the side chains of sugar that interact extremely well with water and also interact with each other quite well. They can also form tangled masses. So in addition to protecting in the stomach the cells from acid, mucuses have very important antimicrobial effects. Mucins are involved partly as passive defenders. Here's a pathogen, bacterium. We see that here's the bacterium or virus blocked by the physical interaction. By the way, this is not to scale. This bacterium should be about 1 micron. The mucuses here would be 1/100th that size. When a pathogen does make it through, contact of a pathogen with mucus, the mucin, the mucus, will tend to stick to the pathogen. That causes a conformational change and makes this mucus be shed. Bacteria that encounter mucus wind up being shed with a coating of mucus, which makes it difficult for the bacteria to survive and reproduce. When a pathogen binds, that triggers a signal transduction cascade that can cause inflammation. It can cause cell death if there's a strong enough signal from multiple bacteria, or it can trigger granule release, more mucus. I'll summarize by running through a simple sketch. Mucin genes are expressed and they wind up in the Golgi. They're processed to have side chains. They may be tethered, they may be dimerized. They're packaged tightly into vesicles that have no water, either in the secreted, to be secreted form or in the membrane-bound form. Those vesicles then fuse with the surface. In the case of the secreted form, those then may form either long sticky strands or tangled meshes of mucus, and the mucus tends to prevent things like acid, pepsin, bacteria, viruses from getting through, either because they're trapped or because the surface membranes, when a bacterium or a virus attaches, causes shedding and the bacterium or virus drifts away completely coated in mucus.